historical description of what's been going on, but actually I'm really most excited about the very last parts, and I'm going to probably skip sections if I need to get there. Okay, so basically, I just wanted to thank the organizers. My attention to this meeting had a sort of strange past because I was both invited and then re-invited. I wanted to especially thank both Shoshana Modak and Dr. Adigonat for their help in getting me re-invited. I was very worried about this invitation because it would have really caused an international situation. Imagine a USA-UK meeting censoring a participant based on their assumed beliefs. This would have been a terrible precedent, one that I think many totalitarian countries around the world would be great to wave in our faces. Anyway, okay, so it's going to be a talk with 6 sections, a bit about the birth of computational biology, applied computational biology, which involves design, the PDB and knowledge-based force fields, quantum mechanical force fields, machine learning, and AI. Okay, so basically between 1950 and 1960, UK and US scientists defined biology as physics. And I think in all of science, it's critical to remember that we all want to stand on the shoulders of giants. And no matter how good we are always doing stuff that's based on the work of other people. There were a number of giants that affected my career. Linus Pauling, who basically found the alpha helix by modeling. Francis Crick, who discovered the structure of DNA by modeling an experiment with Jim Watson. John Kendrew, who basically discovered the method of determining the structure of proteins. And Max Perutz, who in fact was John Kendrew's senior supervisor and Francis Crick's PhD supervisor at the time when Crick did his work with Watson. Can you imagine nowadays if a young person had done something like discovering the structure of DNA, that his supervisor would not put their name on their paper? But the paper really is Watson and Crick, not Watson, Crick, and Perutz. Anyway, these scientists really founded molecular biology, and what did they see? So basically, the secret of all life is a single slide. Information is in the DNA. That information becomes protein structure by a complicated process using the ribosome. Proteins spontaneously fold up. We're going to hear more about that. Once proteins are folded, they have a function, for example, binding the substrate. If that is good for the cell multiplies and the cycle of life continues. So what are proteins? Proteins are basically chains of beads. 20 different kinds of amino acids form a chain. This is a very, very short protein with just 58 amino acids in the chain. And the key thing about proteins are that they self-assemble. Self-assembly is something that we're not really used to. I tell people it's like a contractor would bring a whole pile of bricks to the house, press a button, and all the bricks would decide exactly where they should go. This is how nature works. Self-assembly is critical. And they do this because there is a potential energy, and going downhill is towards the self-assembly, either by minimization shown there, or by dynamics shown here. Now, proteins actually, when they're folded, have this alphabet of colors. In fact, there are 20 different colors, but I just decided to use 5 colors. So some amino acids of the same color. And what's really important is that when the protein folds up, everything is a solid lump in free space, a solid object like you see here rotating. Proteins have this shape, and by virtue of their shape, they carry out their function. So the key thing in protein design is to design a sequence which leads to a shape. This is something that nature decided all by itself. It's the most amazing design principle because once you have a machine that can make protein sequences, the rest happens automatically. Now, each amino acid actually has a color, a different type, and you can see that in free space, different amino acids come close together, amino acids that might not be close together along the sequence. Not only does an amino acid have a color, an amino acid is a group of about 10 atoms, it has a shape. So here is exactly the same thing, showing the amino acids as shapes. Sorry. And that's again rotating. And now you can see that they have either long shapes or some of them are rings. And it's this packing of shapes that essentially is the key to the protein folding problem. We'll see at the end of my talk that you can think about proteins not necessarily as a chain, but as a lot of little clusters of amino acids that like to be together in certain directions. Okay, this is just showing a picture of what protein folding might look like. It's not a real protein folding. It's a very simple protein where by using 2 degrees of freedom and normal modes, we can sort of simulate unfolding and folding. It just shows that the chain generally is disordered, moving around a lot. And then as it folds up, the helices form first, and then there are these strands, and finally it all comes together in this way. Now it's okay. Okay. So basically, that is sort of the summary of biology. What about computational structural biology and empirical force fields? Shoshana told you a little bit about this, but basically I was born in South Africa, came to England in the winter of 1963, and was completely addicted to television. I'd never seen television before. There was no television in Israel at that time, and television just grabbed me. Fortunately, there was only one channel, and the BBC on this channel had John Kendrew, who had just won the Nobel Prize 11 months before, giving a talk on the introduction to molecular biology with programs like the revolution in biology, inside the cell, proteins in 1 dimension, proteins in 3 dimensions, etc. This could still be the basis for even living architecture, the viruses of things today. Now, basically, I decided to think about not to go back to South Africa, to stay there, and actually do a first degree in physics. I was actually very lucky, and you're going to find this word used a lot during my talk. Here, I was lucky to have been born in 1947. And the reason was that it made me a teenager in the 60s. And basically, I was able to read papers that were really quite remarkable. And some of the people who really influenced me, besides Crick, Kendrew, and Perutz, one was David Phillips. He solved the second protein solved, lysozyme. He was a really early supporter of computational biology. Another person that Shoshana also knows, Cy Leventhal, who in 1965, in the same year, actually 66, I'm wrong, in 1966 had an article on computer graphics and protein folding in Scientific American. And then Harold Shiraga, who in 1967 was already doing minimization. He found a new different structure for the alpha helix, and generally was a really early proponent of energy minimization. We'll hear more about Shiraga as this goes on. So basically, Kendrew said that if I wanted to do a PhD in Cambridge, I needed to go to Israel for a year. I didn't really understand why he was sending me to Israel, but he knew that Shneur Lifson, whom Shoshana Warwick already mentioned, was working on the energy functions that define how small molecules interact. And Kendrew realized from his view of myoglobin and of lysozyme that proteins are just small molecules tied together and interacting. Lifson at that time was working with Arya Vash, his student, on the consistent force field. And the basic idea was to have an energy function, potential energy function, describing a molecule as bonds and angles and torsion angles and non-bonded pairs, etc. This is all very, very classical and rather old-fashioned. And you can say that this is Hooke and Fourier. This is really a dead physicist's force function. That function has parameters in it, these Ks and K5s and epsilons, etc. So Lifson and Varshall wanted to take an energy function like that, calculate the properties of small molecules, and then go back empirically and change the parameters to get things to fit small molecules. And my task in this was to write the computer program that for any small molecule would calculate the energy, which is the enthalpy, the structure, the conformation, the vibrational spectrum of these molecules. It turned out that it took about 6 months. I actually didn't think it was very hard. I'd never programmed before, but I don't think I really understood that this was a big deal. So we wrote the program, and basically it turns out if you have an energy function, you can do a great deal. So you can do energy minimization where you go downhill on the energy function. One of the nice things in physics is that dimension doesn't really matter. 1 dimension or 2 dimensions is the same thing, or n dimensions. You can do normal mode dynamics where you oscillate around a minimum, you get in 2 dimensions. You can do molecular dynamics, which is like minimization. It uses the forces, but it has momentum and can go over barriers. And finally, the most recent of these methods is Monte Carlo, developed during the Second World War. And there basically you jump randomly over the surface and eventually end. Now these energy functions, these methods are all much older than the energy functions. Euclid probably was doing minimization. Galileo maybe was doing normal modes with his pendulum. Newton was doing dynamics. And most surprisingly, the simplest method, which was random Monte Carlo jumps, was used to help design nuclear weapons, and was actually classified for some time. That's the Metropolitan method. Okay, so basically we now get onto the period where I am working with Lifson. I have to go back to Cambridge to do my PhD, and this basically introduced multi-scale modeling of macromolecules. I must say it's a term that I never actually used. I just want to show my pointer, sorry. It's a term I never ever used. It was used by the Nobel Committee. It's called flerskalig in Swedish. And I guess it was a way of them sorting out somebody who had worked on more than one scale. But anyway, we worked on the level of simplification. And simplification is very, very important. Einstein has said that everything should be made as simple as it can be, but not simpler. And that is really, really important. It might sound trivial, but actually, if you think about it, every problem has the right level of simplicity. So using the same methods that I used on small molecules, I was able to do minimization of a large protein, or the two proteins that were available, myoglobin and lysozyme, with about 1,000 and 1,500 atoms respectively, using the same energy function. With Aryabhash, after my PhD and during my postdoc, we were able to do a coarse-grained simulation of protein folding, which Shoshana also mentioned, where an amino acid is represented by a lollipop, and you can basically go downhill by minimization, and if you reach a minimum, you jump out of the minimum. Turns out that something that we used back then is actually very important, is the contact map. This is something that the first reference I can find to it is David Phillips in 1970. I somehow remember that it was developed by a Japanese person called Oi, but I couldn't find it. And basically what this is a two-dimensional map where an X or plus marks that these two amino acids are close together. So there's always stuff along the diagonal because amino acid adjacent along the chain are close to each other, but then you get these off-diagonal features. This is the map of the structure very close to the X-ray structure. In our previous folding attempt, we got to about 5 angstroms, a map like this. It looks sort of similar, but it really isn't similar. And this just shows that each of these dots actually corresponds to amino acids close in space. So now you can think of a protein as essentially a collection of pairs of amino acids of certain shapes. And this will end up being very important towards the end. And then Aryabhash also with me worked on a method of adding electrons to calculations so we could look at enzyme reactions. And this was the other side of the scale, more detail, more atoms. Okay. Basically, while we were doing this, simulation was developing. The key person pushing simulation ahead was Anissa Raman, who actually was at the meeting that Shoshana and I attended in Paris in 1976. He sadly died relatively soon after that. He had done a simulation of liquid argon in 1955 with molecular dynamics. And then with Stillinger, Raman had worked out that you could simulate liquid water in 1969. So this basically said if you knew the force field or the energy function, you could simulate the motion. This is what the theory is relatively easy, but I'm not going to go through this. We don't really have time. Basically, Martin Karplus, with his postdoc, Andy McCammon, and PhD student, I think at the time, Bruce Gellin, wrote a paper in 1977 where they simulated the motion of a protein. And this was actually revolutionary because we'd always seen proteins as still, and now we saw them moving. The trouble is that in their paper, this molecule is moving. And how do you know it's moving? Well, look, here the tail is pointing down, and here it's pointing up. So this really said that we needed to use molecular graphics to make a movie. Now, of course, in 1978, when I made this movie, computer graphics was extremely primitive. This movie was made by another really important person in our field, Richard Feldman, who had access to the largest computers at the NIH. Making a movie like this would be trivial on even a 10-year-old phone. At that time, it was very difficult. And in fact, the person who wrote the algorithm to simulate these shaded spheres went on to become one of the founders of the computer graphics company for movies, Pixar, Porter. So this was really important as an early step of visualization. Turns out that all these early calculations were done in vacuum and proteins fold up in water. So it ended up being important, here's Richard Feldman's name, important to actually put water in the calculation. That ended up taking almost another 10 years. It wasn't easy. problem really is that there are many, many atoms in the water. We showed that in water, a protein is much more native-like, that alpha helices can denature in water, and that DNA is also stable in water. So this basically set the scene for the earlier work. I'm now going to jump ahead 30 years, actually, yeah, from 1990 to now, to work we're actually doing now. And this actually involves simulating functional motion. This is on the ribosome, a system that I think Ade or Nat will talk about next. Very large system, has as many residues as even large proteins have atoms, this number of protein residues, this number of RNA residues, and the molecule actually acts in a very simple way. It basically takes in a tRNA comes in as an adapter, carrying an amino acid. It's got to fit. This one doesn't fit, so it gets kicked out. Yep, doesn't fit. Another one comes in, seems to fit, good. This amino acid now moves from the end of the tRNA to join the growing polypeptide chain. Okay, there it happened. And now we continue everything again. Now you see, in order for this to work, the whole system has got to go backwards once it's moved forwards. And we were trying to see what this would really look like in atomic detail. So this is what we did. We looked at a whole lot of different structures of ribosomes, put them all into the same coordinate frame, cleaned up all the coordinates, and then made a movie just showing how the ribosome moves ahead. This is the head of the small particle, the body of the ribosome, one transfer RNA, another transfer RNA, and a messenger. And you can see here that the motion just shown here in large is very, very pretty. Basically, the head rotates. As it rotates, it moves the tRNAs along. This tail becomes close to that, and we can do the reaction. So essentially, it's a kind of rotating head mechanism that seems to account for what really is the key system in life. This is basically the 3D printer that takes the information in the DNA and prints out a protein sequence, which then spontaneously folds up. Okay, so everything I've said so far has been pure science, but of course, biodesign is all about designing things that are going to be useful. So it turns out that early on, very early on in the 80s, I actually was involved in some early biodesign of antibodies. I think it also shows sometimes that things that are not necessarily super interesting scientifically end up being incredibly important for design and for companies. So basically, antibodies are like fingers. You have two hands coming together, 6 fingertips, and those fingertips make different shapes. Here they're making a shape that's round, so an apple binds. If they made a shape that was thin, something else would bind. So how do we actually get these shapes? So nature does this by using proteins. These proteins have 120 amino acids in each hand, if you like. And these hands have fingers. These are these 3 loops that stick out. And it turns out in the mid-80s, Arthur Lesk and Cyrus Chosia were working on ways in which you could predict the structures of these loops. They did it by hand modeling. I joined forces with them and wrote a computer program to do it. And essentially, we published papers in good places in the 80s. But really, I sort of forgot about this until I moved to Stanford in 1987 and sort of got involved in a startup company called Protein Design Lab. And in 1989, that company published a paper, Protein Design Labs. The first author is Carrie Queen, who was the founder of the lab. And here am I. And essentially, this paper has a title that tells the whole story. It's a humanized antibody that binds to the interleukin-2 receptor. Cancer cells have on their surface lots of interleukin-2 receptor. If you have an antibody that can bind the interleukin receptor, you can recognize cancer cells. How do you make antibodies? You take these cancer cells, inject them into a mouse. The mouse makes an antibody. You take the antibody from the mouse, you sequence it, and now you synthesize and clone a synthesized piece of DNA that will then make this protein in monoclonal cells. Turns out that is not a good thing to give to people, because a mouse antibody, even if you make it very pure, will cause an immune reaction in a person. So what you need to do is humanize the antibody. And this is, in fact, something that is done in the computer or on a piece of paper, where you take the mouse antibody and you make changes to its sequence to make it more like a human antibody. This had been done initially by Greg Winter in 1984. Greg Winter got the Nobel Prize, I think, 2 years ago. And his idea was to say as follows: the fingers do the recognition. So let's just take the fingers from the mouse, the hand from the human, this is figuratively speaking, put them together, and now make a chimeric antibody where we have the big part, the gray part from human, and the red fingers from the mouse, and now it will recognize the correct antigen. Cary Queen changed that recipe in a subtle way. He said that you need to also make changes in this intermediate region. Now, why he did that was he saw the model that I built for them. This is just the computer model of this particular antibody. The red are the loops, the white is the body, and these blue residues are residues that are unusual in this region. So this key idea probably would not have been very important scientifically, but it led to a better pattern. Because if you were going to make antibodies with this idea would always do better, because you could make 3 changes that would make the antibody better. In any case, this idea was adopted. It was adopted by large companies. Genentech bought the patent from, or licensed the patent from Protein Design Lab. The patent expired in 2014, but towards the end, Protein Design Lab was getting royalties of the order of $500 million a year. And that's because these antibodies were used to treat cancers. Herceptin, Avastin, and Lucentis, their big sellers, are actually quite well known. And the idea was that we took cells, injected them into mice, and then humanized them using this trick. So it just shows that design can be really, really important, but often you're doing something that isn't that remarkable. I guess it's important, though, that this was done in 1989 and not in 1999, because it was done early enough to really make an impact. Okay, so now let me add more about force fields, and I'm now going to talk about 3 different force fields. One is what I call the PDB and knowledge-based force fields. The first person to do this was Harold Shiraga with Seiji Tanaka in 1976. That's pretty early. And their idea was to actually look at how often different amino acids were in contact in real proteins. By 1976, I think we had, I would imagine, 20 or 30 protein structures were known. So now there was a database in which you could work. And if you saw, for example, that a positively charged lysine is always near a negatively charged glutamic acid, then that would be something that you would use as a long-term interaction. A clever idea. But in fact, Harold Shiraga made one very serious mistake. And this is a warning to all of you. This paper, which was published in 76, has exactly 3 references, all to Harold Shiraga. Let me just tell you that that is a really bad idea. Always refer to all the work no one minds. This was an important advance, but basically in science, you can't rewrite history. Any attempt to rewrite history is discoverable because everything is cited, everything is dated. But it was an important discovery. So this idea has been used often. The idea is simply that you look at a certain interaction. Let's say I'm looking at the interaction between this particular atom, which is the OD atom on aspartic acid, and the CD2 atom on phenylalanine. I will just count how often they occur at different distances apart. And if they occur more often than you expect, it's an attractive interaction. If they occur less often, it's a repulsive interaction. Turns out that what you're seeing here is a slide that is probably more relevant to what DeepMind did than anything else I've talked about, so pay attention. Now, we just simply, people were using this for atom interactions because in order to collect statistics, you generally will only look at a small number of atoms. And what we've seen is that Tanaka and Shiraga, I think, used 10, but they might have used 30. 10 years later, Miyazawa and Jernigan looked at 100 proteins, Sippel again a bit more. Ram Samudrawala and John Molt looked at 1,200 proteins, and we most recently now looked at 10,000. DeepMind has now looked at about 200,000 proteins. The number of proteins is sort of growing exponentially, and the more proteins you have, the more different atom types you can have in this kind of presentation. So for example, when Miyazawa and when Tanaka and Shiraga did it, they might have just simply said, these amino acids are attractive or repulsive. When Samudrawala and Molt did it, they could actually use the individual atom types, and there are 180 atom types in proteins, and so on. Okay. So we tried this, and made an energy function where essentially what we did is, showed to here, we replaced the van der Waals interactions and the electrostatic interactions by a spline fit to a log normal likelihood. So essentially we replaced an empirical function that measured how often, for example, SOD1 liked to be near phenylalanine CD2. And when we did this, we found that actually we could get quite good refinement of protein structures. How did we measure this? Well, we actually generated with normal modes many artificial structures that were near the native structure. And then we took each of these structures and we minimized it. And what you can see if you minimize a structure is that if the energy function is good and your structure is at the right minimum, when you minimize it won't move. But if the energy function is no good and you minimize, you'll go down to the minimum. So basically, if these decoys move towards some point, that suggests that point is a minimum. This just shows this in some detail. These are real structures, and this is the energy contours. And basically, all of these green dots are starting conformations. Zoom in over here, and zoom in over here and over here. Doesn't always work. Here, we started out over here. The native structure's here, but the energy function was wrong, so it moved us away from the right place. And again, this happens in other places. So essentially, this is a way to see how good your energy function is. And by using this method, we could show that the knowledge-based energy function was in fact better than the conventional Coulombic and van der Waals energy function. Now, and this is just showing some of the structures. It's not terribly interesting. These movements are very, very small. It's just used for refinement. Okay. So let me now say something about quantum mechanical force fields. Quantum mechanical force fields actually were around, I think, in the 70s. Enrico Clementi from IBM lab had more computer power than anyone else in the world and was trying to parameterize energy functions by taking small molecules and doing quantum mechanics between them. Trouble is that quantum mechanical calculations take a lot of computer time, and even he didn't have enough. So let's just fast forward to say, firstly, summarize where we are. So basically, I said that simulation started with argon in 55, water in 69, then proteins in water, and many, many, many people are running MD simulations of proteins. We now have at least 1 billion times more resources. In general, the systems have become much larger. The runtimes have become longer, but the energy functions have become no more complicated. We're still using the same sort of billiard ball, electrostatics, spring bonds, energy functions for most simulations as was used before. Is this a good idea? Okay. So in fact, although nothing bad has happened, it does seem that this dead physicist's energy function should be replaced by something more modern. And I did something with this, but not academically, but in fact with companies. It turns out that nobody wants to develop new energy functions because too many people's careers are in running AMBER or CHARM or GROMACS or any of those functions. It's very hard to get public funding for a new energy function. So actually we got it. Algodyne was formed by Carrie Queen in Moscow. He's the person who had founded Protein Design Lab. You saw that Protein Design Lab got a great deal of money from the royalties on antibody design. He used this money to set up a company in Moscow, which basically had a lot of physicists trying to rethink a force field for simulations. This force field would not be Schrodinger's equation. It would not be a real quantum mechanical treatment, but it would be a force field that essentially has electron clouds, exchange, induction, and dispersion. I tell people a force field, a bit like Pauline's book, The Chemical Bond, put into a computer. The company basically stopped in about 2008 and has since been funded again in a new company with many of the same people called Interx that is still ongoing. So basically, what do? We basically, in this case, take, say, 2 water molecules and put them in a certain pose and then another pose, and maybe generate 1,000 different poses for these 2 water molecules. And for each pose, calculate the quantum mechanical energy. And from that, we can see what the interaction energy would be as a function of distance. We can do exactly the same thing with 2 methane molecules. And then to our surprise, when we now look at methane water, the quantum calculation is actually measured by this interaction. So there's a certain level of transferability in these quantum calculations, but essentially it's based on a great deal of parameterization. So the first question to ask is, do we need this? So we tried a simulation where we now replaced the electrostatic term by a quantum mechanical calculation. Now you can see the clouds fluctuating. This was done by 2 former PhD students and a postdoc on I guess the Star Trek Death Star of molecules C60, buxman-fullerene, putting that in water, running it quantum mechanically and classically, and seeing what is the difference. This just shows what we're looking at. The molecule, this is a sphere. We would like to look at the patterns of water around the sphere. The best way to do this is by this map projection. And this just shows where the oxygen molecules are around the sphere. So essentially, we're looking at a surface like this. And we're going to look at the surface at different distances out. We're going to do this classically and quantum mechanically. So this is very, very close. This is the quantum mechanical calculation. You can already see an imprinting of the shape. This is just showing the shape. Now we're going to move out. You're going to see basically, this is the radial distribution function where my mouse is. And what you're seeing is that basically there is much, much more structure quantum mechanically than there is classically. And basically quite soon, we're at 8 angstroms, at basically 9 angstroms, there is essentially no structure classically, but there is still some long-range structure quantum mechanics. So the polarization is making the water molecules more structured. Not a big surprise. Okay, good. And this just shows that when you get to 10.4 angstroms, there's still some structure there quantum mechanically, but nothing classically. Now to the final part, machine learning and AI force fields. Okay, so basically, we've all seen the fanfare that appeared on the DeepMind blog about 2 weeks ago. I must say I actually missed it because I was so involved in coronavirus that I can't talk about, but still, my postdoc who's been working on protein folding sent me the recording, and yesterday I sat down and listened to it in great detail, and quite frankly, I was blown away. So basically, DeepMind, what did they really achieve? And there's a very nice blog that was written from Oxford, PIG, Protein Informatics Group, by Carlos Ortega Rivera. And he basically describes their result, and it really is a very large team of people. Here we have on the slide, I think there are 19, but there are probably altogether 20 or 30 people involved. They have more resources than everyone else added together. They're backed by Google and a lot of money, but in fact, the results are really spectacular. I did not expect to live to see this, and we can see this here. This is a distribution of the sum of z-scores of the predictions. And of course, the sum is if you do more predictions and you do better predictions. So the best group got a z-score sum of 210. The 2 second best groups, 93. So it's more than twice higher. So, and of course, you can realize that for a competition like CASP, where you have to predict unknown proteins, the amount of manpower you have is very important. If you have a much bigger group, then each person can spend more time. If you have much bigger computers, and in fact, one of the winning groups was Baker, who has a lot of resources. Another winning group who tied was Zhang, and he has rather limited resources. But the important thing is that with enough resources, DeepMind was able to do something really amazing. And what is surprising is they did better in all categories. This is just showing, so CASP has been improving over the years. The first CASP, I think, was in 1992, but I'm not completely sure, maybe 1994. And I was there. But basically, back in those days, the quality, how well you were doing, shown here on the y-axis, against the difficulty of the target, people could do well on easy targets, but very badly on difficult targets. As we move forward 20-something years, the groups at CASP, not including the best group, can do this well, but the best group does this well. So essentially, the progress here has been quite remarkable. And the question has to be, what have they done? Now, I must say, I have generally been skeptical of deep learning because I've always felt that it's a black box. We don't really know what's going on inside. So let's look at this in more detail. So basically, like all science, DeepMind is based on previous work, but machine learning depends more than other science on previous work because it actually needs examples. So there were many thousands of experimentalists involved using X-ray crystallography, NMR, and cryo-EM to solve examples of structures. There were even more molecular biology and bioinformaticians determining sequences of proteins by either the old-fashioned protein sequencing before 1985 and then DNA sequencing. And there were thousands of theoretical chemists and physicists developing the methods and finding the best representations. Now, DeepMind totally acknowledges the fact that they use all this information, and that's fine. They're doing exactly the right thing. So basically, DeepMind predicts structure really well. They called it alpha predict, but I think it should have really been called AlphaFold, because folding is a physical process where you start with an unfolded chain, and then you optimize some energy function and make it fold. So basically, protein folding is a physical process. The other thing is that in these predictions, DeepMind, as well as all the other contestants, use multiple sequences. They use sometimes thousands of sequences for the same protein, and the sequences end up giving you important information. But real protein folding is a simulation. It doesn't have to be done by simulation. We did it way back in 1975, but the answer was simply not accurate enough. Okay, so basically, I would say that one person who's contributed most to the science behind DeepMind is Chris Sander. In 1977, Chris Sander actually came to do a postdoc in Israel with Shneur Lifson, continuing the Lifson tradition. With Shneur Lifson, he published this paper in 1980 where they were looking for specific recognition between amino acids in protein tertiary structure. This ended up being something that was very, very hard to find. A lot of people were very skeptical, but basically if you fast forward, I guess 31 years to a paper again involving Chris Sander, this time with Deborah Marks, they actually found that you really could look at multiple sequence alignments and find correlations that suggest that certain amino acids are actually close together in space. This was a very important breakthrough. And if we go back and look at CASP, you'll find that there was a sudden jump. This is where that idea came in. And suddenly the worst structures did much, much better because using many sequences, we could actually see correlations between sequences that indicated closeness in space. Basically, Chris also used multiple sequences with Burkhard Rost and machine learning back in '93 to basically take a multiple sequence alignment and predict secondary structure. This is now being done very commonly, and their method is the forerunner of HH-Pred or Pred-HH that is also used by DeepMind. Okay, now let's just see what DeepMind did. So this is the first thing I thought was very clever. In a neural network, you have all these different cells that are connected to other cells. But the trouble is if you looked at a protein, then every single protein amino acid can affect every other protein amino acid. So if you have a protein of 1,000 long, you're going to have 1,000 times 1,000 objects. And these can all be affected by sequences and so on. So the idea that they had was to basically try to group cells together, group learning units together that actually correspond to the graphical structure of the protein. If two amino acids are close together, then their units should be close together. Now, you don't actually do this by physically changing the machine. You just simply have a flag that tells you that certain units are going to be looked at. And basically, by doing this, we're able to get rid of a lot of the noise because units that shouldn't be there are not allowed to talk to each other. Okay, so that was very important. So basically, they have an end-to-end pipeline. And what this really means is that a multiple sequence alignment goes in, all the structures go in over here, and out comes the structure. At CASP 13, 2 years ago, they actually put out a function, a potential function that ended up being optimized on in a normal way to give a structure. The good thing about an end-to-end pipeline is that the method is aware of the result and can actually estimate the confidence of various areas. And this, I think, was very, very important. Okay, but they're essentially learning from input sequence alignments and input fragment structures. Okay, things are going to get cleverer. So basically, they don't just put into this amino acids. which if you like is what Tanaka and Shiraga did, what others after that, they actually put in chunks. The chunks could be templates, groups of amino acids, even large bits of protein. And we'll see how they do that. So essentially, they're able to take what they call templates, and these templates have a structure, they have contacts, they have sequences. They're treated as rigid objects. And these are residues of fragments of proteins. Fragments have been used before. Fragments were used by Sander, they were used by me in 1970. They're very much the basis of Rosetta, and it's very clever. But doing it in this way of machine learning is particularly clever. Okay, so this is what they then did is, because they have fragments that are chunks of templates, they also convert every single amino acid into a fragment. They break the bonds connecting amino acids. And this is again very clever because it means that we have the same unified treatment for any rigid object. The rigid object could be a domain that you already folded and so on. Okay. And then their network also sort of keeps track of itself so you can actually see how it's thinking. And, you know, I really hope that this powerful method will in fact be able to work by putting in one sequence and not all the sequences, or even perhaps by putting in or knowing how to take a sequence and generate a matrix, looking at how easy it is to modify certain sequences. Okay, so just to sort of try to recap, and I'm going to try and go back to one of my earlier slides, which really doesn't matter. If you think about a protein, not as a chain, let's see here, think about this not as a chain, but as a collection of amino acids. These amino acids are pairs. We know it's a collection of atoms and the atoms are pairs, but the amino acids are pairs, but they're pairs with lots and lots of degrees of freedom. The amino acids themselves have different numbers of atoms, they're flexible, and they have different orientations. But machine learning is perfectly designed to remember all of this and to remember it in a way that doesn't involve overlearning. It can also remember not just pairs of amino acids, but clusters of amino acids. So now it's a bit like doing a jigsaw puzzle by learning what pairs like to come together. And you know, if the pieces are unique, you'd get the answer immediately by that. If the pieces are not unique, you would try different combinations. There's a lot to try, a lot of computer time, but I think this algorithm where they basically treat these rigid units either as amino acids or as larger units, and they're able to assemble them is indeed very clever. So I think we probably are going to see a solution to the real protein folding problem. It's also very important to realize that there's a lot of, in this competition at CASP, the groups are all getting better. I mean, even you know, the groups, each year things are getting better and everyone is learning from everybody else. So it's actually a very, very good sign. So just to end up, I wanted to mention that we talked about these different things, really a very wide-ranging talk. I have currently a rather small group, but we are having fun, doing very different things. And I have been supported by the NIH for 30 years, and I'm very grateful to that. Zhao just did some work on COVID. Ivan is still trying to fold proteins and so on. Okay, Andrea is also working on COVID. Thanks you all very much indeed. Thank you.